Hey guys, my name is Tieho and I'm Tutorbox's life science tutor. Yay! Welcome to Tutorbox live in conjunction with COVID-19 catch-up sessions. So if this is your first time joining us since our first session, I'm going to introduce myself again just in case, because you don't know who I am. So first of all, I am not 14. I may look 14, but I'm not 14, okay? A lot of people say that I am 14 years old, but I am an oldie, yet a kid at heart. I study biomedical engineering at the University of the Witwatersrand, and this year is my fourth year. Whoop whoop. It's been a journey, guys. It's been real. So, I love many things, okay? Among those many things is biology. I love bio because it's just so interesting having to, study how living organisms live, how they interact, how they breathe, how they move, how they eat. It's just very fascinating. Like every single time I go through like my notes and stuff like that, so every single time I learn something new in class, it's just so fascinating. Just like, what? That happens in us? What? That happens in other living organisms? What? It's just very interesting and very intriguing, getting to know how we operate, how we move, how we all do this living thing, you know what I mean? And for me, it all just reflects like God's wonderful work that He has done, on earth. So yeah. I decided to tutor bio because, believe it or not, I was once upon a time in your position. I used to struggle with bio, and I never used to get like the master I wanted to get, and I didn't understand why until I started doing bio again when I started in university my first year, and I just noticed how many things I missed out on when I was in high school, and I'm like, oh, if I could have paid attention to this, if I could have paid attention to that, then I would have done much better, you know. But that's why I'm here to help you. So fear not, we are going to get this, all these little tiny things, because bio builds on. From grade 10, it builds on to grade 11, from grade 11, it builds on to grade 12, from grade 12, it just goes on into university and forevermore in your life. So we need to understand the basics before we can understand the complicated things. Okay, so what we'll be looking at in this lesson is that we're going to do revision on the structure and function of the cell, and we'll also look into different types of cells, and then afterwards we'll finish off with mitosis. All right, so as I have said before, the cell is the basic unit of life. All living organisms are made up of cells. There are unicellular organisms like bacteria, and there are multicellular organisms like us humans. So cells differ in size and in shape, okay? And they differ like this because they have different kinds of functions that is related to their size and their shape. So we have some squamous, some cuboidal, some columnar, even some hexagonal shaped cells. Okay, so now let's just relate the shape of the cell to its function. So squamous cells are those thin kind of cells. They basically deal with what do you call it? Protection, and they act as a diffusion kind of barrier, if that makes sense. So basically whatever they line, they control what enters and what leaves the organ, okay? So for instance, if we're looking at the lungs, okay, we can find squamous cells in the lungs. What they do is that they allow for gaseous exchange because they are very thin, okay? And yeah, so they allow for the movement of gases. They allow for the movement of some electrolytes. So in your blood vessels, you'll find sodium, potassium crossing over the the blood vessels into the extracellular space. But yeah, that's just more depth work that you don't have to know right now. But just have that idea that diffusion happens across these squamous cells because they're thin. Okay, and then cuboidal cells, what they do is that they mostly function in secreting substances. So you'll often find these types of cells in glands. They produce whatever kind of thing that they're supposed to produce, and then afterwards it secretes it into the organ that is at. So your pancreas that has cuboidal cells, okay, your sweat glands that has cuboidal cells. And then lastly, we have columnar cells. The columnar cells are basically for absorption. Okay, and some of them, like, have these specializations on their apical parts, like on top of it. They have these villi. Okay, and what these villi do is that they are responsible for, like, absorbing nutrients and, like, absorbing other substances that the body needs. So you'll find a lot of these in your digestive system. So more specifically, like your intestines going onwards. Okay, so these are the shapes that I was talking about: the squamous, cuboidal, and columnar. You don't really have to know like the hexagonal one. That's gonna come up, like, later in your life if you do histology. But anyways, yeah. So these are just the types of cell shapes. Okay, so what I'm going to do is I'm going to draw out all these organelles. I'm going to explain to you, like, what each of them do. So it's essential, like, in biology, for you to like draw out things and like label them and stuff like that as you go along, because that helps you to learn things better. Okay, instead of just sitting down and reading your textbook, draw it out, try picture it yourself, because one day there could be like label the cell or draw the cell, and like you miss an organelle or two because you don't actually, like, take the time to draw it and stuff like that. Like, some of us can't just cram by looking at textbooks. You have to actively do the work. So let's get to it. Okay, so let's start off with our structural organelles. Okay, so we have the cell wall. Okay, so basically what the function of the cell wall is to provide, like, that rigid shape, and it's supposed to protect like the inner parts of the cell. Okay, so we have the cell wall. Now we're going to add in the cell membrane. Okay, here is our cell membrane. So if we take a deeper look at how the cell membrane is composed, we will see that it has a phospholipid bilayer and it has proteins in, like, the layer. Okay, so let's just quickly draw that out. Whoops, there, mine went skew a little bit. Okay, we'll add in our protein. Okay, so here we have our phospholipid bilayer. Okay, so these over here, your phospholipid, and we have two of them. Okay, so phospholipid bilayer. Okay, so we basically call this model of the cell membrane the fluid mosaic model. Why? Because of two things, right? It's in its name itself. So the fluid part comes from the functionality of, like, the phospholipids. What the phospholipids are is that, well, the way they're joined, they're not rigid, okay, like the plant cell, wall is. But this over here is not rigid, so it's able to move and stuff, and it, like, basically, if the cell, like, experiences a change in like temperature and stuff like that, what the fats do is that, like, if it's too hot, then they tend to, like, separate from each other, and if it's too cold, then they come towards each other. Okay, so that's why, like, we call it a fluid. That's what the fluid and the fluid mosaic model represents. And mosaic would just be because it's got all these compositions. So it's got the proteins, it even has cholesterol between the lipids. We got, like, the phospholipids here. It's just, like, made up of a combination of compounds. Okay, so fluid mosaic. Okay, so basically the function of the cell membrane is to control what enters and what leaves the cell. Okay, so there are two modes of transport that happen across the cell membrane. We have active and we have passive. So active transportation is just basically the type of transportation that requires energy, okay, ATP. So this usually happens in cases whereby you have things that are traveling against their concentration gradient. So for instance, right, let's just say that this side is the outside of the cell and this side is the inside of the cell, and I'm the cell membrane. My head is the cell membrane. Okay, and I have a lot of sodium on the outside and I have a little bit of sodium on the inside. So now I want to pump the sodium that is on the inside to outside, okay, even though there's a lot. So we're going to need some energy to push sodium against its transportation gradient, okay? And then passive transport. So we're looking at diffusion and osmosis. So basically this is transportation that doesn't require energy. So like sodium, for example, can travel across the what do you call this, the cell membrane easily just by diffusion. And this happens because, like, let's just say again, we're at the outside of the cell and this is the inside of the cell. So sodium, because there's a high concentration over here and a low concentration over here, sodium is going to travel to the place where there is a low concentration because it wants to balance out the what do you call it, wants to balance out, or wants to be at like an equilibrium state, like, across the walls. So the same thing happens with osmosis, is what happens with water molecules. So if the concentration of like sodium is too high on this side and we have some water here, what it's going to do is that it's going to diffuse through so it can like separate it. Okay, so next up we have the cytoplasm. Okay, so what the cytoplasm does is that it's like, well, first of all, it's like a jelly-like kind of substance, okay? And what it does is that it provides the cell, like, with some shape, okay? And also what it does is that it provides like a space for all the organelles to dwell in, and it allows some biochemical like processes to happen in it. Okay, so next up we have the vacuole. Okay, so the function of the vacuole basically, like, it's large in a plant cell and small in an animal cell. So it's large in a plant cell because what do you call it? Like it provides that structure in the what do you call plant cell. It helps to provide that structure, and also it's like, very tense. So like it gives it some tough volume, if that makes sense. And then in both the plant and animal cell, what it does is that it's a storage place for some dissolved substances like salt, sugar, and proteins. And what else it can do is that it can store waste. Okay, so next up we have the nucleus. The nucleus is the boss. Okay, so I'm going to draw like a large image of the nucleus so we can see, like, what's going on it. So here we have our chromatin network. And then afterwards we have that little dark ball that you find, like, in some scans, electron microscopic scans. So we call this dark ball the nucleoid. Okay, and then we have our nuclear membrane that has pores in it. Okay, so basically what the function of the nucleus is that it stores the genetic information. So it stores your DNA in it, okay? And what your DNA does is that it encodes, like, for the processes that happens in the cell. So it deals with protein synthesis, and it deals with other kinds of cellular responses that the cell would do. Yeah, so basically your DNA, it contains your hereditary information and it contains your genetic code. Okay, and then last but not least, inside this space of the nucleus, we have nucleoplasm. So it's basically like cytoplasm, but inside the nucleus, nucleoplasm. All right, so now from the nuclear membrane, we have this organelle called the endoplasmic reticulum. So the endoplasmic reticulum is like the membranous extension of the nucleus. Okay, and basically, like, the endoplasmic reticulum can be divided into two. So we have the smooth endoplasmic reticulum and the rough endoplasmic reticulum. So the rough indicates that we have ribosomes on them, and the smooth doesn't have ribosomes on them. All right. So the function of the rough endoplasmic reticulum is that because it has these ribosomes on them, it's responsible for producing proteins. And the smooth endoplasmic reticulum, that one produces, like, important lipids that are needed for the cell. So maybe it could, like, produce the lipids that are needed for the cell membrane. You can look that up. But yeah, that's what the functions of these two are. Okay, so next up we have the Golgi apparatus. Okay, so the Golgi apparatus is an organelle with membranous folds, okay? So it's got that kind of like appearance. And basically what its function is to do is, its function is to process the proteins that are made, and then afterwards it packages them into little vesicles so that, what do you call it? These vesicles will basically, like, travel either along the cell and take the protein to a specific region, or these vesicles will leave the cell and go to another cell. Okay, so next up we have the mitochondria. What I'm going to do is I'm going to draw it the same way I did with the nucleus and stuff on the outside the cell, so we can see what it is composed of, its structure and stuff. Mitochondria. Okay, so basically the mitochondria has got the outer membrane and it's got the inner membrane. So the inner membrane has got these little extensions called cristae. Okay, now also in the mitochondria we find some ribosomes in it. Okay, and in addition we also do have some genetic information that is stored inside the mitochondria. So fun fact, the genetic information that is stored in the mitochondria is the one that's from the mother. So your maternal DNA is found in your mitochondria, and apparently people can trace back your ancestry history by studying the DNA that is in the mitochondria. Pretty cool, huh? So yeah, so your mitochondria has a lot of ribosomes in it, and the ribosomes, what they basically do is that they're responsible for producing enzymes. So remember that the ribosome is the powerhouse of the cell, okay? And a cell could have many of these things scattered across the cytoplasm, okay? So now you need these ribosomes so it can produce these enzymes, so it can help to make the process of making energy faster. Okay, so basically what the mitochondria does is that it takes in oxygen and glucose and then it converts that into ATP, and that ATP goes and it gets used in the cells. Can you name one place that I spoke about earlier where ATP is needed? I'll wait for an answer. Okay, so next up we have lysosomes. Lysosomes are small little organelles, and basically what their function is to do is to break down protein. So old protein, damaged protein, and stuff like that, they just break that down. Okay, so the last organelle that we'll be looking at is the plastids. Plastids are only found in plant cells. So we have three types of plastids. We have chloroplasts, chromoplasts, and leucoplasts. Okay, so basically, I'll explain just going backwards. So leucoplasts are basically colorless organelles. Okay, and they're usually found in regions where there is no sunlight. So for instance, the roots. Okay, so basically what they do is that they store food in different forms. So what they do is they store starch, lipids, proteins in the roots of the cell. Then we have chromoplasts. Chromoplasts are basically yellow, red, and orange in color, and they're mostly found in plants that are bright in color. So we're looking at flowers, looking at fruits, and some leaves. So yeah, these colors could be to attract some insects and other kind of organisms. Okay, the last thing that we have is chlorophyll. So basic not chlorophyll, chloroplast. So chloroplasts contain chlorophyll in them, hence it has that green color. Chlorophyll is a green pigment. Okay, and basically what this thing is responsible for is that it traps sunlight, or rather just uses the energy from the sunlight so that it can convert that energy into ATP, so that the cell or rather the plant will have some energy in it. Let's go with the cell. The cell will have energy to do its function. So let's draw what a chloroplast looks like. Okay, so a chloroplast has a double membrane, and then in it we have these little lamellae. Okay, but these lamellae are found in stacks. Okay, and in the lamellae we find the chlorophyll, and a whole stack of lamellae is called a granum. And lastly, we have this a little ball of starch, if I can say it, not a ball, but just starch that is found in the cell. So basically, it's going to use the starch for energy. And then afterwards, we also have ribosomes that are around the cell. So these ribosomes, they produce enzymes so that the cell can use so that the enzymes can fasten the process of creating energy. Okay, so let's just go through some basic questions that they could ask you, like, in a test or exam. So when it comes to, like, cells, they love asking questions where you compare, like, the difference between a plant cell and an animal cell. Okay, so you can do the obvious stuff. You'll be like, a plant cell has chloroplast, it's got a cell wall, large vacuole. Okay, and then afterwards you just say, like, the animal cell lacks that, lacks that, lacks that. Okay, and then afterwards you can just go and list the difference, like, from then onwards, any more that you can think of. But those are the basic ones that I always used to go to. Okay, and a lot of the times they'll also ask you to label. Okay, and or first, like, they'll put up a plant cell and an animal cell, and then they'll ask you, is this a plant or animal cell? And then you have to say why. Okay, so it's kind of the same as, like, the thing that I just said before, when you have to compare the differences. It's just that you're going to say, okay, fine, this cell has ABC. Okay, now a lot of the times they also like to put on electron micrographs up, and then they ask you to label it and stuff like that. And sometimes it can be, like, a little bit confusing, so I'll just go through one of them with you right now. Okay, so here's an electron micrograph of a plant cell. How do I know it's a plant cell? I can see that this cell has a double membrane. Okay, and it also has these organelles that I can see clearly that I wouldn't find it in an animal cell, because that is the chloroplast. Okay, and just a little tip. So a lot of the times, like, if with electron micrographs, if, like, a substance is stained white, then it could be, like, some kind of liquid, or it could be a fat. Okay, so in this case, I know that the vacuole is like a liquid substance that stores the waste and everything else that I've said before, and I know that is present in a plant cell also. So I would label B as a vacuole. Okay, and so just looking at the other ones, so the dark circles that are just around. Okay, so I would label that as vesicles. So was that C would be a vesicle. So why I would say that, because I know that vesicles, they package proteins, and a lot of the times proteins and other acids are, like, stained in this darker color. They appear black in electron micrographs. So here, because I can see that there are many of them and it kind of looks like they're migrating out of the cell, then I would label them as vesicles. Remember, vesicles are packaged by the Golgi, and they package, and then they are, like, taken out of the cell, or they can be taken to some part of the cell for some kind of process. Okay, so let's see what else. We have E. A lot of the times also, they'll point at something that you'll be able to identify. So E looks like a rod-shaped kind of organelle. And a rod-shaped organelle that I know of is a mitochondria. So I would label that as a mitochondria. And then, okay, so A would be cytoplasm, B would be vacuole, C would be vesicle, D, like I said, I saw that as a chloroplast. How do I know? When we drew it out, we know that we had these lines that just, like, they stretched out through our diagrams and stuff. So that's how I would label this organelle over here. That's how I would identify this organelle as a chloroplast. So what would be in it would be chlorophyll, you see. And then F would be the cell wall, the most outer part. You can see that the arrow is clearly pointing at the outer part. That would be the cell wall. The cell membrane is on the inner side. And then G, I would say that that is like the chromatin network, or I'd also say chromosome. But double check because of the way that it appears. Just double check, like, with your teachers. And then H would be the nuclear membrane. I can't see what I is pointing at, but J would be, can you see the cell membrane? Okay, so a lot of the times they will ask you to label it, and then they'll also ask you for a function of what you labeled. Okay, so just be aware of those. Okay, so now we are going to head up into our break. So if you have any questions, please can you post them in the Q&A section, and I'll be sure to answer them. Thanks, guys. Enjoy your break. welcome back. Now we're going to continue on with mitosis. So we're going to be looking at all the phases that are involved in it, what its importance is, and then furthermore we'll be looking at like the disease that is related to mitosis. All right. So mitosis is basically the process whereby a cell, we'll call the cell a parent cell, divides into two daughter cells, okay? And each of these daughter cells will have the same number of chromosomes in them, okay? So it's basically like, cellular division in a way. So yeah, the function of mitosis is that it is supposed to replace damaged and what do you call it, dead cells. It's for growth of tissues, and also for asexual reproduction. Okay, and the phases that are involved in mitosis are what do you call it, prophase, metaphase, anaphase, and telophase. Okay, interphase is not a phase in mitosis. Basically, what happens in interphase is that this is the phase where the DNA is replicated, where it's copied. Okay, and interphase basically happens more in the cell cycle. So interphase is just basically involved in the cell cycle. So basically what happens in interphase is that the DNA is checked before the cell goes into mitosis. So a cell is only allowed to replicate a specific amount of times, okay? This is done to prevent what do you call it, damaged DNA from being copied, and then it results in the formation of tumors and stuff like that, which is like cancer. But I'll get more into that later. Just know for now that interphase is involved in the cell cycle, and we have prophase, metaphase, anaphase, and telophase that we're going to focus on now. Okay, so let's draw this out. The best way to learn is to draw things out, okay? So the first phase that happens is interphase. Interphase is where DNA replication occurs, and this is the longest phase, and this all happens before mitosis. Okay, now we have prophase. In prophase, the nuclear membrane starts to disappear or dissolve or whatever it is, okay? The nucleolus, that one also starts to disappear. Your chromatin network starts to thicken and become condensed. And when it does that, it gets your chromosome, because it is doubled, we get this formation of chromatids, sister chromatids, okay? These two chromatids are held together by centromeres, okay? Last but not least, we have our centrosomes. The centrosomes migrate to opposite ends of the pole of the cell, okay? So let's draw that out. And let's just say that we have an organism that has 4 chromosomes. So our chromatin network starts, or rather let's just draw the dissolving nucleus first. Nuclear membrane. Okay. Then our chromatin network starts to thicken. Okay, so now we have drawn out the chromosomes that have thickened, and the chromatin network that has thickened, and then it forms these chromosomes, and these sister chromatids are held together by the centromeres. And then we have the centrosomes that migrate to opposite ends of the pole of the cell. Right. Okay, now that prophase is done, we're going to go into metaphase. Metaphase is when all of the chromosomes line up at the equator of the cell. Okay, our centrosomes are at the opposite ends. Basically what they do is that they release their spindle fibers and they attach onto the centromeres of the chromosomes. Okay, so that is the end of metaphase. So it's just basic chromosomes, they align themselves in the middle, and then the spindle fibers attach into the centromeres of the chromosomes. Cool. Now anaphase is just basically when the spindle fibers contract, okay, and then they pull the sister chromatids, what do you call it? Yeah, they pull the sister chromatids apart. So basically, a chromosome is going to go across, another chromosome is going to go across, okay? Okay, and then after this happens, we go into telophase. So now telophase, basically the nuclear membrane starts to appear, the nucleolus starts to appear, the chromatin network starts to form again. So basically the chromosomes loosen and the DNA just spreads out again into the cell. And then afterwards, what's going to happen is a phase, is a not a phase, but like just call it a phase, called cytokinesis. Okay, so in the animal cell, what will happen is that the what do you call it? The size of the cell will pinch. Okay, and then afterwards it would separate. But in a plant cell, a plate forms in the middle, causing it to divide. A plate forms in the middle, which will then cause it to split into two. Okay, so now we have our two daughter cells that will be a resultant from this division. Okay. So now we're going to look at a live action video of mitosis. Okay, so the disease that is related to mitosis is cancer. So cancer can be defined as like the irregular growth of tissues, okay? And a lot of the times, like remember when I was busy describing interphase and like how it's involved in the cell cycle? So this is also just like a little bit of extra information, but basically what happens is that so your cell is only allowed to replicate a specific amount of times, right? Now, how this is controlled is that like your cell like has a timer, if I can say. So basically, on your DNA, at the end of like of the strands, both ends, we have like these telomeres, okay? And these telomeres become shorter and shorter every single time DNA replication occurs. Okay, so the shorter they are, yeah, so it reaches like a specific length. When it reaches that length, then the cell cycle comes to a stop. The cell cycle will be like, okay, fine. This cell is not allowed to divide anymore. It can stay and like still produce whatever or like function as whatever it's supposed to do, or it will have to die. Okay, so now with cancer, what happens is that it bypasses like all of these laws of the cell cycle. Okay, so what do you call it? Basically, like what happens is if your DNA is damaged in any way, or like if it has some mutations on it, then what it can cause is cancer. Okay, and cancer doesn't happen like in what do you call it, every single individual, because I mean if you look at what the causes are, it's radiation, it's like eating processed food, it's smoking, all these things, like a lot of people do them, but a lot of them also don't have cancer. Okay, so even though like those stuff do cause cancer, it doesn't like immediately result into cancer. Okay, I mean like we're exposed to sunlight every single day, and like some of us more than others, but we don't have like skin cancer, for instance. So yeah, basically, if the DNA is damaged, okay, then that can like lead to a cancerous a cancerous cell like occurring, and then that cancerous cell divides and it becomes so many that like it forms tumors. Okay, so now when a tumor is formed, we have like two different types of tumors. Okay, so we got benign tumors and we've got malignant. Okay, so benign tumors are basically tumors that are non-invasive. Okay, so meaning that like they don't they form, but like they don't grow into your other tissues and stuff like that. But malignant tumors are the ones that like are like cancerous, in that it spreads its cancerous cells throughout the whole entire body, and it does this because like it invades into the tissues. So when it invades into the tissues, some of the cancerous cells can end up in like the blood vessels or the lymphatic system, and that travels throughout the whole entire body. And when it does it, like that cell can stop in some region, then will start like multiplying and forming tumors, and like another one will form somewhere else, and another. So it just starts like spread throughout the whole entire body. Okay. And so basically, treatment for cancer, you can cure, or cancer can be cured depending on like what stage you catch it at. So tumors that are like benign, okay, those can be treated with by surgery, okay, by surgically removing them, so cutting them out, okay. So things like breast cancer, that can be cut out. But cancers that are like malignant, the ones that have like spread throughout the whole entire body, those I would think like that radiation and chemotherapy would be like the best kind of way to cure them, I guess. But okay, no. So radiation, basically what they do is that they use radio waves to destroy the cancerous cells, okay? And even though radiation does cause cancer, if like you use it to if you target it at the cancerous cells, it can kill the cancer cells, okay? And chemotherapy, basically like a person has to take oral medication that's like that has like chemicals that can kill these cancerous cells, okay? But they are like well, rather these treatments kind of like have negative impacts on people, and that like makes them lose their hair. They can lose some body functions and stuff. They could be tired all the time and stuff like that. So yeah, it's rough. And also what we learned in varsity was like we also like studied about cancer, and like there were some interesting ways that people are trying to cure cancer. So for example, like they use radio not radio what do you call it, lasers, to treat cancer cells. So basically what they do is that they use the heat to burn up these cells, you know. And also another thing was that we learned was that so what cancers do is when they're in that the benign stage, okay, what they do is that they grow to a certain size, okay, and then afterwards it stops growing. And what it does is that it releases some kind of hormone so that it can induce the nearest blood vessel to like what do you call it, grow another like vessel, so it can come into that tumor and feed that tumor with more like food that it needs to be able to grow. Okay, so we learned that they are trying to like make some medication against that what do you call it, that hormone. So yeah, it was just kind of like very interesting. Cancer is like a very interesting study, so like if you guys are interested, get into it. It's very interesting. But yeah, that's just about it. Okay, so some famous mitosis questions. A lot of the times they like to ask, or what they'll do is they'll put like photos of different stages of mitosis, and then they'll ask you to label what stage they're in. Secondly, they will ask you to like label certain structures that are found in that stage of mitosis. And then thirdly, like they could ask you what the function is, okay, of like whatever it is they pointed at. Okay, so that's number one. Number two, a lot of the times like they'll ask you to count the number of chromosomes that are found inside that nucleus. Both of these are a chromosome, okay, the one that stays by itself and that, those are both a chromosome, okay. So don't get confused. This here has got what do you call it, chromatids. Each individual part is called a chromatid, and if it's just by itself, it's just a chromosome. The both of them together, which is cute, the both of them together, that is a chromosome, okay. And then a lot of the times, like they'll have a phase of mitosis, and then they'll ask you to draw out the next phase, okay, or like explain what's going to happen in the next phase, okay. And then further on, I think that's it with mitosis. Those are the most important stuff that I can remember. And then with cancer, they'll ask you what was the cause, the treatments. Like they'll ask you like what kind of treatment is better. So like I said, or like I was describing before, the radiotherapy and the chemotherapy, and like the surgical one, they'll ask you like, is it efficient to just surgically remove a tumor and like why or why not. But this also depends on the stage of cancer that the person is in, so watch out for that. And yeah. Okay, so this is the end of our session. Thank you so much for tuning into this Tutorbox live in conjunction with COVID-19. I hope that this was resourceful. I hope that this was helpful for you. I hope that just learning the little things will help you to bring the bigger things together, like what I was really saying earlier on. Okay, so please be sure to do the exercises that we provide for you guys. And also, if you have any questions, please can you post them down in the question and answer section, and yeah, I'll be sure to answer them. Thanks, guys.